Hello. Hello, dears. Am I audible? Can I please get the answer for that? Am I audible or am I visible here? Hello, yes. Good evening. Good evening, everyone. How are you all? Did you have class today? Yes, okay. How was the class? I'm good. I'm good. I'm really good. Thank you for asking, Niranjana. How was the class today? Good. Okay. So your exams are coming up, right? Your term exam is coming up. So yeah, so are you all prepared for that? All the chapters for this examinations, is it over? Are you all prepared for that? Oh, exam is over. Okay. So how was the exam? Good, okay. Yeah. Okay, so you also had a session yesterday, like Vivek sir was there, and was that class helpful? Or are these sessions helping you for your preparation for the exam? Yes, okay. So we'll have a quick revision of today's chapter, that is fundamental unit of life. Okay, this is the very first chapter. Oh, I forgot to introduce myself. I'm Anna. So I'll be dealing with you this chapter. Okay, fundamental unit of life. Okay, good evening, everyone. Okay, so this is a very first chapter. You know what a cell is. Yeah, you all have a basic knowledge of what a cell is, right? Okay, so in this chapter we are going to cover what about the structure of a microscope and the unicellular and multicellular organisms. What is the basic difference between them, and some examples for this unicellular and multicellular organisms, and what is the basic difference between a prokaryotic cell and a eukaryotic cell. It is very simple, right? And the structure, what a cell is made up of. Okay, what are the components inside a cell? Like plasma membrane, cell wall, and how the substances get transported across a cell. Okay, how the substances or the gases transport or move from one cell to the other or the adjacent cell, and about some cell organelles. Very few cell organelles are discussed in this chapter, right? Okay, so we will just start with basic experiment here. Okay, so what do you know this experiment, or have you heard of this experiment regarding the cell? We'll be taking, so to start with, we'll be taking a small piece of onion. Okay, we'll be taking a small piece of onion, and we'll be just taking off the peel. Okay, the skin of the onion from the inside. We'll be taking a small peel or the small part of skin from the onion using a forceps. Okay, using a forceps. Can you see here? Okay. Okay, using a forceps, we'll be taking out a peel. We'll be taking out a peel or the skin. Okay, and like our aim is to observe the cells. Our aim is to observe the cells. What are the components inside a cell? Okay, and we'll be placing that in a slide or on a glass slide, and we'll be observing. We'll be adding some drop of saffron in. What is the use of saffron in? What is the use of saffron in? Do you know what is the use of saffron in here? Yes, very good. Staining. Okay, I want a complete response from your side. Okay, so saffron is used for staining. So otherwise, we cannot observe the cell more clearly. Okay, color. Yes, very good. Very good. Color. Yes, to stain that. Okay, so while we are observing through the microscope, we will be getting a very simple specimen or very simple, like we will be getting a image. Okay, and before going to that, let us just discuss what are the parts of a compound or a light microscope. Okay, in the textbook there is just a figure only, so there is not any single explanation regarding this parts of a microscope. We can just deal with the parts of microscope now. Okay, so the very first part is the eyepiece. Okay, eyepiece. Through the eyepiece, we are just looking into onto the specimen. So this is the very first part of a compound microscope. And coming towards the bottom, we can see an objective lens. So this is the lens through which we can like see the image. Okay, the objective lens here. Now we have there are two adjustment systems in this microscope. One is fine adjustment and the other is coarse adjustment. What is the difference between this coarse adjustment and fine adjustment? What is the difference between the coarse adjustment and a fine adjustment? Have you ever observed any specimen through microscope? Have you ever observed? No. Okay. So yeah, you'll be having practicals. Okay. You'll be having practicals. So surely you will observe a specimen through a microscope. Okay. And there are two main focusing parts for a microscope. Okay, there is one coarse adjustment and there is a fine adjustment. So coarse adjustment is through a coarse adjustment knob, we can just initially focus the specimen, just initial focusing only. Okay, in a low magnification, just initial focusing only. But when we rotate the fine adjustment knob, we can clearly, specifically see the cells. Okay, this is a fine adjustment, that is with a higher magnification. Okay, did you understand this? Coarse adjustment and fine adjustment knobs. Did you understand that? Yes, okay. So these are the two adjustment knobs present in the microscope. Coarse adjustment and fine adjustment. And then here you can see a stage where the specimen is placed. Okay, where the specimen is placed, and there is a condenser here. What is the function of a condenser? What is the function of a condenser? Okay, here there is a light source. There's a mirror here on this light source. When the light is reflected on this light source, this light will be going to this condenser, and this condenser focus this light onto the specimen. Okay, this condenser is actually placed below the stage. Okay, here you can see the stage where the specimen is placed. Okay, and the condenser is present below the stage. Okay, the function of this condenser is to focus and direct the sunlight to the specimen so that we can see the image, or we can see the specimen more clearly. Okay, so these are the basic parts. Okay, there are some small specific parts here, but you are going to deal with these parts only. Okay, you should study these parts only. Eyepiece, objective lens, fine focus, coarse focus, there's a stage, condenser. Okay, is it clear? Is it clear? Okay, yes, very good. Now, while we are observing this specimen through this microscope, you can see, okay, you can see an image like this. You can see an image like this. This is the image of the onion skin. Okay, what is this actually? What is this called? Or what are the each components or each compartment here called? Each compartment here. Yes, very good. Very good, students. You're always responding to my questions. Okay. Okay, thank you so much. So, cell. So, this is called a cell. Okay. So, we are going to deal with the properties of cell. What is a unicellular organelle? What is a multicellular organelle? Everything in this chapter. So, I will try to touch each and every portion of these chapters. Okay. So that I will not miss any single portion. Okay, for the exam. So there are two types of organisms based on the number of cells, based on number of cells in an organisms. The whole organism is divided into two basic categories. One is unicellular organism and the other is multicellular organisms. Okay, unicellular organism and multicellular organisms. Okay, so the examples of unicellular organisms are shown here. Okay, one is amoeba. It comes under protista. Okay, amoeba that comes under protista. Just study these examples. Okay, paramecium that also comes under protista. And here you can see bacteria. Okay, here you can see bacteria. So this bacteria comes under kingdom Monera. You need not study these kingdoms. Okay, you will be studying that later. But I'm just sharing you an information here. And Euglena comes under Protista, and Chlamydia single cell organism are known as unicellular organisms. Very good, Hassan. Okay, Chlamydomonas, it also comes under Protista. Okay, so these are unicellular organisms. Okay, these are the examples of unicellular organisms. What are unicellular organisms? The organisms that consist of a single cell. They do not have multiple cells. So the examples are amoeba, paramecium, chlamydomonas, euglena, bacteria. Okay, now the next category is multicellular organisms. So what do you mean by multicellular organisms? What do you mean by multicellular organism in contrast to unicellular organisms? More than cells. More than cells means? More than how many? Multi. Okay. More than? Yes. More than one cell. Okay. The organisms that consist of single cell is called unicellular organism. But the organisms that consist of more than one cell, they are known as multicellular organisms. We humans, me, you, everyone, we are multicellular organisms because our body have different types of or different kinds of like cells present in our body. Okay, we have skin cells, we have nerve cells, we have blood cells, we have a lot kind of cells. So these are called multicellular organisms. Examples are fungi, plants, and animals. Okay, so these are multicellular organisms. I'm messing up that organelles and organisms. Okay, so these are shown. The shown cells are different kinds of cells present in a human body. Okay, human body. Different kinds of cells present in a human body. We have nerve cells. What is the function of nerve cell? Do you know this function of your nerve cell? What is the function of nerve cell? Anyone can say. Transferring messages. Very good. It helps in the control and the whole coordination of our body to transport messages from the brain. Very good, Srija. Helps us to react to stimulus. Yeah, there are different stimulus. When we touch a hot pan, we get a stimulus there. So we can sense our nerve cells sense that stimulus. Okay, so that we can withdraw our hand from there. So it helps in the control and coordination of our body. And we have blood cells, another category of cells. Smooth muscle cells. We have muscle cells. Then fat cells are present. And bone cell. The bone present in our body. It is a type of cell. Okay. Then in females, there is ovum. And in males, there is sperm. So these are different kinds of cells present in a human body. Okay, transporting. Very good, very good. So these are different types of cells present in a human body. Okay, is it clear? Is it clear? Yes, okay. So now we will do a question. Okay, here there is some question. Match the following. Please, okay. Match the following. Here there is a match the following present. So there are different scientists shown here. Robert Brown, Purkinje, Robert Hooke, Schleiden. Okay, I will just say some of the scientists and what they have done in biology or in with regards to cell. Okay, first we will deal with Robert Hooke. Okay, Robert Hooke. What Robert Hooke done was that he discovered cell, found cell in 1665. He discovered cell. Okay, and back to Robert Hooke, there was Leeuwenhoek. Leeuwenhoek. He actually discovered free-living cells in pond water. Okay, he discovered free-living cells in pond water. That was done by Leeuwenhoek. And another scientist was Robert Brown. Okay, so what Robert Brown did was, after the discovery of the cell by Robert Hooke, this Robert Brown, he actually had a keen observation in the cell, and he found out nucleus. Yeah, I am getting loads and lots of answers here. Nucleus in the cell. And another scientist was Purkinje. Purkinje, actually, he coined the term protoplasm. Actually, what is protoplasm? What is protoplasm? What do you mean by protoplasm? Or what is the difference between a protoplasm and cytoplasm? Okay, here there are two terms we are discussing in this chapter. Protoplasm and cytoplasm. Okay, yeah, very good, Hassan. Okay, so while we consider a cell, okay, there is a cell membrane. The contents or the component present inside the cell except the cell membrane, they are called, or that is called, protoplasm. Okay, that means it includes the nucleus also. Here there is nucleus. So this is cytoplasm, the fluid filled part, including the organelles. There are some organelles present here. Okay, so the cytoplasm plus nucleus, all the components inside a cell, it is called protoplasm. Whereas cytoplasm is protoplasm minus nucleus. Protoplasm minus nucleus, that is the fluid filled substance including the cell organelles other than nucleus. It is called cytoplasm. Is it clear what's the difference between protoplasm and a cytoplasm? Yes. Okay. Okay. Yes. Very good. So protoplasm is the it refers to the whole components present inside a cell, including the cytoplasm and nucleus. But cytoplasm is protoplasm minus nucleus. Okay, so Purkinje coined the term protoplasm. Okay, and another two scientists were Schleiden and Schwann. Schleiden and Schwann. They formulated the theory called cell theory. So what is cell theory? What is cell theory? Cell theory just simply says that all the animals and plants are made up of cells. That means we are made up of cells. Plants are made up of cells. Every organism is in the universe is made up of cells. All the plants and the animals are made up of cells. Okay. And another thing was the cell is the basic unit of life. Okay. So these are the 2 points that is being discussed in cell theory. All living organisms are made up of cell, and the cell is the basic unit of life. Why is it called basic unit of life? Why is it tagged as basic unit of life? Yeah, like before forming an organism, actually the cell itself is formed into tissues, and they itself is formed into organs, and the building part is done by cell. Okay, that is why the cell is called the building blocks of the cell, or it is a fundamental unit of cell. Yes. Okay. So Schleiden and Schwann, they formulated the cell theory. Okay, is it clear? Is it clear? Okay. Clear. Okay, very good. And there was another scientist named, okay, just a second. Okay. Okay. There was another scientist named Virchow or Rudolf Virchow. He actually expanded the cell theory. And he said that all the cells existed or all the cells are formed from pre-existing cells. There is a cell and that cell divides into different cells and the new cells are formed. That's what Virchow said. Okay, so he simply expanded the cell theory formulated by Sridhar and Schwann, and he said that all the cells arise from pre-existing cells. Okay, is it clear? Now you can match the following, right? Okay, okay. So Robert Brown, what did Robert Brown discovered? Tell me, what did Robert Brown discover? Yes, very good. Nucleus. Then what did Purkinje did? What did Purkinje did? Protoplasm. Very good. He coined the term protoplasm. What did Robert Hooke did? Yes, he discovered cell. And what was done by Schleiden? He formulated cell theory. So A2, yes, B4, C1 and D3. So this is the answer. Okay, is it clear? Can I move to the next part? Yes, okay. Now, why is light microscope called a compound microscope? Why is light microscope called a light microscope is called a compound microscope? Why is it so? Why is it so compound? Why is it so? Do you know? Actually, the reason is that it has two types of lenses that work together to magnify an object. We have seen that already. I showed you objective lenses, right? So there are two objective lenses, and these objective lenses, there is an objective lens as well as a ocular lens. Okay, here I will show you. Okay, here you can see an eyepiece. Here you can see an eyepiece, and also here you can see objective lenses. So there are two types of lenses present, and these two lenses actually they coordinate together and form the image. So that is why the light microscope is called the compound microscope. Understood? So it has two types of lenses that work together to magnify an object. Yes, the ocular is the lens closest to the eye, whereas the objective is the lens closest to the object. So that work together and form what? Compound microscope. Okay. So now the next question is, do you agree cell is the building unit of an organism? If yes, explain why. Yeah, I have already told you about that. Cell is the basic unit of organism. So do you know the level of organization of an organism? Levels of organization of an organism. Can you say the levels of organization? Cell is the building unit of an organism. Okay. Like if we build a house, we know that we use bricks, right? So these bricks are combined together, okay, and we create or a house is formed there. So likewise, cells is also the combined together and it makes up an organisms. So there is a level of organization here. The simplest unit is cell, and the cells combine together to form tissue. Okay, to form tissue, nervous tissue, for example. And this tissue, they again combine to form an organ. Okay, organ. And this organ again, like they club together and form an organ system of our body. And finally, an organism. So this is the levels of organization of an organism. Okay. So that is why it is said that cell is the basic unit of an organism. Is it clear? Is it clear? Yes, okay, very good, Sriraj. Now, we'll be moving on to the next main important part, that is plasma membrane, what a cell is made up of. From outside we can see a plasma membrane. So there is lot to do with the plasma membrane. Okay, actually there is lot to do with the plasma membrane. It actually protects the cell from its external environment. So this is a cell. Here there is a plasma membrane, and this plasma membrane separates the external environment and the internal environment of a cell. Okay, it acts as a barrier, and it allows, the main function of the plasma membrane is that it allows the transport of selected substances only, into the cell and towards the outside. Okay, so it allows the transport of selected particles or selected substances only. That is why this plasma membrane or cell membrane is also called selectively permeable, because it only selects some of them. Selectively permeable membrane. Okay, and when we come to the structure of the cell membrane, it is made up of a lipid bilayer. Okay, it is made up of a lipid bilayer. They have two layers of lipid. That is why it is called a lipid bilayer. And the round portion of the spherical portion is called hydrophilic part. That means it is water loving part, and the tail-like part is called hydrophobic part, that is water hating part. Okay, so this is the basic structure of a plasma membrane. Okay, now here you can see it, the molecules transportation here through this plasma membrane. Okay, and another main advantage of the cell membrane is that it ensures a flexibility, or it has, this plasma membrane is very flexible. Okay, and the structure of plasma membrane is actually set through fluid mosaic model. Why it is explained as fluid mosaic model? Because of its flexibility itself. Okay, because of its flexibility itself, the plasma membrane is called fluid mosaic. Okay, so this is the basic structure of a plasma membrane. Now, this is a question. How do substances, I have already said, this plasma membrane allows or permit selected particles to move across the membrane. But how does this movement takes place? Or how do substances move across the cell? Okay, so for that, we will just look at a scenario here. So what is the difference between, you said it right, Lakshmi. Okay, so here there are two scenarios. What is the difference between these two scenarios? Or the first scenario and the second scenario? Which is the hardest scene here? Which is first or second? Which is more harder? Or which requires more energy? Here there is a rock and he is going to pull the rock. Here he is going to push the rock. So which is hardest one? The second part, yes, the second scene is the hardest one. This is more hard, or it requires more energy. Okay, it requires energy for the transport. Okay, it requires energy for the transport. So this is called active transport. Whereas the transport that does not require any energy, just pushing, just here he just pushed that only. He didn't go like go behind that rock. Okay, so here there is no energy required. So that is why this is passive one. Okay, so there are two types of transport here: passive transport and an active transport. So these are the two types of transport that takes place across the cell membrane. But here in this chapter, we are going to deal with the passive transport only. Okay, so there are two types of passive transport that we are going to deal with. That is, one is diffusion and the other is osmosis. Okay, is it clear? Can I move to the next one? Can I move? Yes. Okay, so the first process is diffusion. Okay, what you can see here, what is diffusion actually? What is diffusion actually? What is diffusion? It is a movement. I know that. Yes, it is just the movement of substances. Yes. Actually, yeah, I'm getting different responses. Okay. So actually, like here we can consider a beaker of water. Okay, a beaker of water and a small concentration of dye. Okay, this is a dye and this is the beaker. Okay, so the concentration in the dye, the concentration of solute here is higher, right? And here the solute concentration is lower. Okay, so when we give or when we put a drop of dye in this water solution or water in a beaker of water, what happens after some time? This spreads throughout the water, right? This dye will be spreading towards the water, to the whole water. And after some time, if the dye color is red, the whole solution will be turned to red color, right? This is a case of diffusion. So here what happens is that the solute particles or the substances move from a higher concentration to a region of lower concentration. High to low. More specifically, we can say in case of solute. The movement of solute from a region of high concentration to a region of low concentration. Did you understand that? What is a diffusion? Like when I studied in my back in school days about this diffusion osmosis, I was very confused. Okay, I know you will be also a little bit confused about this diffusion and osmosis, but this is it. Okay, try to study it simply. Try to study it with more examples. Okay, can you quote another example of diffusion? Can you say another example of diffusion other than this dye? Can you say from our day-to-day life? Like you all sometimes will be using that thing. Spraying perfumes. Yeah, you said it right, Hazan. Spraying perfume. Okay, when in a room, when we spray the perfume, now, after we sprayed the perfume and we kept it there and we left the room, and just after a few seconds we came back to the room, the whole room smells this perfume. Why is it so? Diffusion takes place in the room. The perfume molecules or the molecules present in the perfume or the particles present in the perfume diffused in the air. Okay, diffused in the air from a higher concentrated area to the lower concentrated area. So this is diffusion, a passive transport. Is it clear? What is diffusion? Is it clear, dears? Yes. Okay, so this is diffusion. And what about osmosis? And what about osmosis? Is it the same as that of diffusion? Or is it different? Actually, this takes place in respect to water molecules or the solvent molecules. Okay. So the basic difference between the diffusion and osmosis is that in osmosis, water molecules or the solvent molecules move from higher concentration to a region of lower concentration. But there is a specific thing, that is through a semi-permeable membrane. Through a semi-permeable membrane. That is the most stressed part of that. Through a semi-permeable membrane. If you write the answer of what an osmosis is, if you didn't write the write that it takes place through semi-permeable membrane, you won't get the mark. Okay, so stress on that. It is the movement of water molecules from a region of high concentration to a region of lower concentration through a semi-permeable membrane. Okay, you can give an example for osmosis now. Can you give an example for osmosis here? Can you give? Yeah, everyone is thinking of that. Sometimes you might have seen your mothers or father doing this thing. Example is, yes, dry raisins when put in water. Dry raisins. Yeah, pickling mangoes in salt water, raisins in water. Yeah, I'm getting too much response here. Drying fruits, everything. They are examples of osmosis. So what happens here is that we are keeping, okay, here there is a cup of water and we are putting the dry raisins here. Okay, we are putting the dry raisins and after 24 hours, what will happen? What will happen? This dry raisins or the shrunken raisins, they got swollen. What happened here? What actually happened here? The water molecules, the concentration of water was higher in where? In the solution. Okay, so this here what took place is that the water moved from the region of higher concentration to the region of lower concentration. Inside the dry raisins, there was not water. So the water entered there. Okay, and the dry raisins got swollen. Okay, here you can see an example, an illustration also here. Here you can see the water and these are the solute molecules here. And this is the solvent part. This is the water part. Okay, blue color is the water part. And after osmosis, the level through a semi-permeable membrane, the water molecules moved from the region of higher concentration to the region of lower concentration. And here it went up. Okay, so this is osmosis. Did you understand that? Do you understand? Yes. Okay, can you give me another example of, you have already given so many examples. Yeah, I'm happy. Okay, so this is the difference between diffusion and osmosis. And based on the concentration of solute, there are three types of solution: hypotonic solution, isotonic solution, hypertonic solutions. Okay, hypotonic, isotonic, and hypertonic. Okay, these are all parts of osmosis. Okay, hypotonic solution, isotonic solution, and hypertonic solution. So what is hypotonic solution? In hypotonic solution, the concentration of solute is less. Hypo is less. Hyper is high. Okay, think like that. Hypo less, hyper is high. Okay, and it is based on the solute concentration. Okay, here in hypotonic solution, the concentration of solute is less in the solution. Okay, so here you can take a cup of water. Okay, here you can take a cup of water and consider this is a cell. Okay, consider that this solution has less solute concentration and more of solvent concentration. More of solvent concentration. Okay, and what will happen here? The water will move from outside to inside and it causes swelling. Okay, it causes swelling. So water moves into the cell and the cell expand and it means lyse also. Lyse means burst also. Okay, did you understand what is hypotonic solution? Can I move to the next one? Hypotonic solution. Yes, okay. So in hypertonic solution, the concentration of solute is less. That is why, and then if the concentration of solute is less, that means the so-called concentration of solvent is more. So the solvent will move from the region of higher concentration to the region of lower concentration here to the cell. And the cell will expand and lyse. Okay. And in case of isotonic solution, iso means same. The concentration here and outside and inside is very similar. Okay, so no overall movement will take place. The overall concentration are equal and the cell remain constant. There is no change in size of the cell. And what about hypertonic solution? What about hypertonic solution? Here, the solute concentration is higher. Okay, the solute concentration is higher. And what will happen here? If the solute concentration is higher in solution, what will happen? That means the solvent concentration here in the solution is less. And the solvent concentration here in the cell is higher compared to the solution. So the solvent moves out of the cell or the water moves out of the cell, causing the shell to shrink. Do you understand that? What is the difference between hypotonic, hypertonic, and isotonic solution? Every one of you? Do you understand that? Yes. Can I get more answers? Did you understand? If you have any doubt, you can ask me now. Okay? No doubt. Okay. Okay. So that means you are clear about this hypotonic, hypertonic, and isotonic solution. Okay. Now, the structure which forms a barrier between protoplasm of the cell and its external environment in an animal cell is, what is that? I want the answers here. Yes, very good. It is plasma membrane. It is plasma membrane. Okay. Now, the next question is differentiate between diffusion and osmosis. Give example. We have already seen what are some examples. Okay, what's the difference between diffusion and osmosis? In case of diffusion, what happens? The solute particles move from a region of higher concentration to a region of lower concentration, and it is a passive transport. And in case of osmosis, what happens is that the water molecules move from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. Okay, that is the most stress part here. Okay, if a plant cell is kept in a hypotonic solution, the cell will, what happens? Okay, this is a cup of water. If the plant cell is kept in a hypotonic solution, here this is hypo, that means the solute concentration is less here and the solvent concentration is higher here. So the solvent will move from here to here, right? And it increase in its volume. The cell will increase in its volume. Yes, all are giving the correct answers. Okay, increase in its volume. Now, what happens to the dry resins when placed in plain water after some time? Yeah, we have already seen that. When dry resins placed in water, what happens? It will, I want the answers. It will bulge or it will get swell. And what would happen if these resins are placed in concentrated solution? So now I have a cell which is swollen up. Now I have a cell which is swollen up. Okay. And if I put this in a salt solution, what happens? What happens here? That means the solute concentration is higher here. That means this solution is hypertonic. What will happen? The solvent will move out of the cell and it will cause the shrinking of the cells. The shrink in a concentrated salt solution. Very good. Okay, I got the answer from everyone. So, this is it. Okay. Now, the most abundant material in plant cell wall is. So, now we are coming to the next part. We have covered plasma membrane. The next part is plant cell wall. Okay. So, cell wall is actually okay, it is not lipid. Okay, it is not lipid. Yes, very good. The other answers are correct. So, in plant cells, there is another covering that is called a cell wall. Here you can see a cell wall. Here you can see cell wall here. Okay, so compared to animal cells, plant cells have an additional covering. Plant cells have an additional covering, and this is called cell wall. And the main component of cell wall is cellulose. And the main component of cell wall is cellulose. And there is some another phenomenon that takes place in plant cells, that is plasmolysis. Okay, what is plasmolysis? From the word itself, it is clear. Plasmolysis. The lysis, the rupture of plasma membrane or the shrinking of plasma membrane. Okay, so what happens here is that, or protoplasm, yeah, shrinking of protoplasm away from the cell wall. Yes, Hassan, you said it right. Okay, so when a plant cell loses water by osmosis, when a plant cell loses water by osmosis, what happens here in plant cell is that the components inside the plant cell, here this is a cell wall, okay, and these are the components inside the cell wall, and this bigger part is vacuole here, okay. This bigger part is vacuole actually. So the components inside the plant cell, they will shrink away from the cell wall. They will detach away from the cell wall and they will shrink. That is known as plasmolysis. Okay, shrinking of protoplasm away from the cell wall. So this is a phenomenon that takes place in plant cells. Okay. Now, how a prokaryotic cell is different from eukaryotic cells. I have put actually questions from each and every topic in this chapter. Okay. So no points are missed here. How a prokaryotic cell is different from a eukaryotic cell. You can say the basic difference between a prokaryotic cell and a eukaryotic cell. What is it? Can you say prokaryotic and eukaryotic? Pro means false. U means true. Karyon is nucleus. So you can say now. Yeah. For a prokaryotic cell, the nuclear membrane, there is a organelle called nucleus. Okay. Where our genes and chromosomes are present. Okay. This nuclear membrane, or there is no a well-defined nucleus. But in a eukaryotic cell, there is a well-defined nucleus. That is a basic difference between prokaryotic cell and a eukaryotic cell. Here you can see the parts of a prokaryotic cell and a eukaryotic cell. So this is a prokaryotic cell, this is a eukaryotic cell. Actually, when we see a prokaryotic cell, here you cannot see a nucleus as that of eukaryotic cell. Eukaryotic cell have a specific nucleus, but here you cannot see a nucleus. Instead, the DNA is actually scattered in a, or it is concentrated to a particular area only, and that area is called nucleoid. That area is called nucleoid. Okay, nucleoid. So this is nucleoid is present in actually prokaryotic cell. There is no specific nucleus structure present in prokaryotic cell. And another basic difference between a prokaryotic cell and a eukaryotic cell is that this prokaryotic cell lack the cell organelles like mitochondria, chloroplast, etc. Okay, but a eukaryotic cell has that. Okay, and these are the two basic difference between a prokaryotic cell and a eukaryotic cell. And here in prokaryotic cell, you can see actually a cell wall here. But this is an exception. Prokaryotic cells have a cell wall. Okay, this is a cell. Here you can see a cell wall. And also a eukaryotic cell called plant cell is actually eukaryotic. Okay, and this plant cell also has cell wall. Fungi has cell wall. Okay. Bacteria has a cell wall. Okay, so these are the basic difference between a prokaryotic cell and a eukaryotic cell. Here you can see this difference. Prokaryotic cells are generally small in size. The image you just saw, here you can see that small in size, but eukaryotic cells are larger. And a well-defined nucleus is absent in prokaryotic cell. And the nuclear region is not surrounded by a nuclear membrane. But in eukaryotic cell, a well-defined nucleus is present. Okay, and in prokaryotic cell, only a single chromosome is present, but in eukaryotic cell, there are more than one chromosome. In humans, for us, we have 23 pairs of chromosome. We have 23 pairs of chromosome, but when considering a bacteria or while considering a prokaryotic cell, that they only have a single chromosome. Okay, so this then a membrane-bound organelles such as mitochondria, plastids, endoplasmic reticulum, Golgi apparatus, lysosome, etc. are absent in prokaryotic cells, whereas in eukaryotic cells these membrane-bound organelles are present. And cell division takes place by fission or budding. They just split into two, and that is how cell division is taking place. But in a eukaryotic cell, there are specific processes called mitosis or meiosis. The cell division takes place through that processes only. So these are the basic difference between a prokaryotic cell and a eukaryotic cell. Okay, so I'm moving to the next part. Here is the question. Membrane-bound cell organelles are not found in dash cells. Are not found in membrane-bound organelles. Yes. Are not found in prokaryotic cells. Yes, very good, very good. Everyone is getting the answers. So, what are cell organelles? Write the names of different cell organelles. Okay, so we know that cell organelles are membrane-bound structures. Okay, cell organelles are membrane-bound structures to make the machinery of a eukaryotic cell easy, because eukaryotic cell for us human beings, we in our body there are a lot complex like metabolic activities taking place in our body. Right. So in order to cope up with all that metabolic activities, we need specialized structures. We need specialized membrane bound structures. So these specialized membrane bound structures are called cell organelles. Write the name of different cell organelles. Yeah, you can say that. Mitochondria. Yeah, plastids. Vacuoles. You can name in the chat box. Vacuole. Yeah. Endoplasmic reticulum. Yes. Ribosomes. Yes. Nucleus, etc. Okay. Lysosomes. Yes, I'm getting it. Now, Golgi apparatus. So these are some of the examples of cell organelles present in a cell. Okay. Here you can see various cell organelles. Now, which organelle will you associate with the elimination of old and worn out cells and why? Elimination of old and worn out cells and why? Okay. This is a process taking place in our body. These are, the answer to that is lysosome. Okay. The answer to that is lysosome. Okay. So lysosome is actually known as suicidal bags of a cell. Why are they called suicidal bags of cell? Are they going to suicide in the cell? Actually, yes. Actually, yes. Okay. This lysosome actually cleans up. Okay. The lysosomes actually cleans up the cell by digesting the worn out cells or by digesting the damaged or foreign particles that is entering into the cell. Okay. That is what lysosomes does. Okay. Here, what happens here is that if consider the cell is damaged or the mitochondria is damaged. Okay. And we need to remove that mitochondria. So lysosomes comes there. Okay. This lysosome attaches to the plasma membrane of the cell. Okay. Okay. I'll repeat. Okay. I will repeat. Okay. Lysosomes is actually called suicidal bags of cell. Okay. Lysosomes is actually called suicidal bags of cell. Why is it called so? That is what I'm going to discuss. Okay. What happens or what is done by lysosomes is that it actually cleans the cells. This lysosomes actually cleans up the cell. Okay. Well, for example, when we sweep our room, okay, you all are sweeping or you all make your room clean in some day or the other, right? So we are just sweeping with the help of a broom. And we are sweeping out all the dust or the unwanted things to the dustpan. And we are keeping or we are putting that in a dust bin. The same process is actually happening here. Okay. So if consider the mitochondria is a damaged particle. We don't want that. Okay. We don't want that. So the lysosomes comes into play here. And the lysosome combines with the cell that contains this damaged mitochondria, and these lysosomes actually has some digestive enzymes present in them. They have some digestive enzymes present in them. And these digestive enzymes enter in the cell, and this digestive enzymes break this mitochondria into small pieces. That is how this mitochondria is disrupted. So the cell is cleaned. Or if the cell itself is damaged, so this is case of organelle, okay, or the cell, if the cell itself is damaged, okay, here there is lysosomes, right? And this lysosome actually bursts itself. This will burst itself, okay? And the digestive enzyme degrade the cell itself, and the cell is gone from that body. Okay, so this is why the lysosome is called the suicidal bag of cell. Okay, did you get it? Someone have asked me to about repeat about lysosomes. Did you get that? Did you get it? Okay, so moving to the next. Now, there was another, yes, okay, thank you. Identify the organelle which can make proteins in cell. Identify the organelle which can make proteins in cell. Which organelle makes proteins inside the cell? Before moving on to that, we will discuss about the another organelle, that is endoplasmic reticulum. Okay, endoplasmic reticulum actually contains loads of a network of tubes, etc. Okay, lots of network of tubes. And there are two types of endoplasmic reticulum. rough endoplasmic reticulum and smooth endoplasmic reticulum. What is the difference between rough endoplasmic reticulum and a smooth endoplasmic reticulum? What is the difference between rough endoplasmic reticulum and a smooth endoplasmic reticulum? In rough endoplasmic reticulum, there are certain structures, there are certain organelles called ribosomes attached to their surface. Here you can see ribosomes. This is an endoplasmic reticulum. Here you can see some bodies called ribosomes. Okay, so these ribosomes are attached to the surface. That is why they are called rough endoplasmic reticulum. Okay, and what about smooth endoplasmic reticulum? Here you can see smooth endoplasmic reticulum. Here there are no ribosomes attached to their surface. So these are called smooth. Their surface appears smooth because of the absence of ribosome. Okay, so that is why it is called smooth endoplasmic reticulum. And what is the function of this rough endoplasmic reticulum and smooth endoplasmic reticulum? Actually, this rough endoplasmic reticulum, there are ribosomes. Okay, ribosomes actually manufacture proteins. Ribosomes help in the manufacture of proteins. And here in smooth endoplasmic reticulum, there is no ribosome. And it helps in this manufacture of fats or lipids. Fats or lipids. And some of these fats or some of these lipids and proteins, some of these help in the formation of cell membrane. Okay, some of these help in the formation of a cell membrane, and that process is known as membrane biogenesis. And sometimes there will be question like which organelle is associated with membrane biogenesis. So what will be the answer? Endoplasmic reticulum. Specifically, the endoplasmic reticulum. Okay, so this is endoplasmic reticulum. So ribosomes, actually, you know that this helps in the manufacture of proteins. Sorry. Okay. Name the cell which do not have a well-defined nuclear region. Okay, membrane biogenesis is what I have told now. That is the synthesis of membrane. Just the synthesis of membrane. Okay, biological synthesis of membrane with the help of the proteins and fats made by ribosomes. Okay, or made by endoplasmic reticulum. It is membrane biogenesis. Okay. So this is ribosome. Describe the role played by the ribosome. Why are they termed as suicidal back? How do they perform their function? It's quite clear to all of you. Role played by ribosome. So these lysosomes are called suicidal back. Why are they called suicidal back? Okay, why are they called suicidal back? They actually cleanse the cell by digesting the unwanted materials or the unwanted particles that enter into the cell, or the cell itself can burst with the help of lysosomes. That is why it is called suicidal back. Okay. Now, how does amoeba obtain their food? Actually, amoeba is a unicellular organism. How does amoeba obtain its food? It is not actually given in very detail in the textbook, but you can see just what happens in amoeba. Okay, through endocytosis. Very good. Through endocytosis. This process is called endocytosis. This process is called endocytosis. I'm taking 5 or 10 more minutes. Okay, so and a foreign body or the food for the amoeba will when the bacteria comes closer, this amoeba, okay, it has an irregular structure. These structures actually forms like a leg, and these are called pseudopods. So you can see pseudopods. It extends like a leg, and it takes the bacteria into the cell. Okay, it takes bacteria into the cell, and the food vacuole combines with this bacteria or the food, and now the food is digested inside the body. Okay, so this is endocytosis. Why is this called endocytosis? Because it is taking into the cell membrane, takes the food material into the cell. Okay, that is why it is called endocytosis. And that is amoeba engulf the food. That is known as endocytosis. Okay. Now, name the 2 organelles we have studied. Okay, that contain their own genetic material. We have studied that their own genetic material. What are the organs? Yeah, I will like stop this now. Yeah, I know you have a master class. Okay, so name the 2 organelles we have studied that they contain their own genetic material. What are they? Mitochondria and chloroplast. So there are some other cells or organelles known as plastids. There are 2 types of plastids: chromoplast and leucoplast. So one type of chromoplast is chloroplast. Here there are pigments called chlorophyll. So, and also they have genetic material of their own. Here they have genetic material of their own. Okay. And mitochondria, they help in the production of ATP or the energy for the metabolic activities that takes place inside our body. Okay. And now we know that cell organelles known as nucleus is present. So the inside the nucleus, there are specific structures called chromosome. For eukaryotic cell or for human beings, how many pairs of chromosomes are present? 23 pairs of chromosomes are present. So what is actually a chromosome? What is actually a chromosome? It is a condensed form of DNA. It is a condensed form of DNA. Actually, DNA is a quite longer structure. And this DNA is actually wind together with the help of some proteins known as histones that you will deal with in later classes. So histones will be helping into the for the condensation of DNA, and it forms chromosomes. So these are DNA and a specific segment of DNA that codes for a function, for the hair color, for the eye color, for the nail structure, for our skin color. So it like wide variety of characters are there. Okay, so a segment of DNA that codes for a function is known as a gene. Understand this? You understand, everyone? Okay, actually the time is up. Yeah, you have a master class now. Yeah. Okay. So is there any doubt regarding this chapter? Is there any doubt regarding this? I think I have completed almost all the organelles and all the contents in this chapter in this short 1 hour. Like it was a bit confusing, and I hope all these contents are clear for you. No, ma'am, it's clear. Okay, thank you so much. Thanks a lot. And all the best for your exams. Okay, all the best for everything clear. Thank you so much. So, all the best for your exams and thank you so much. Have a great night. Sleep well and have a lot of good dreams. Okay. Thank you so much. You can leave. Thank you. Thank you so much. Thank you. I hope this class is very clear to all of you. Bye. Bye. Bye.